In this video, we will look at how protein antigens are processed to allow for adaptive immune responses. I will describe antigens, epitopes, and two types of major histocompatibility complex proteins. An antigen is a molecule that can specifically bind to an antibody. Antigens can be lipids, polysaccharides, or most often, proteins or glycoproteins. The latter are proteins combined with sugar molecules. Exogenous antigens are from outside the body and its cells. These include toxins, secretions, and parts of bacteria, fungi, viruses, and other microbes. Endogenous antigens come from inside the body's cells, including intracellular pathogens such as viruses. An epitope, also known as an antigenic determinant, is a specific portion of or shape on an antigen. Most cells have membrane proteins called major histocompatibility complex proteins, or MHC, that bind and display epitopes on the cell surface. There are two classes of MHC. All nucleated cells use MHC class I proteins to display epitopes from endogenous antigens. Only certain cells of the immune system, called antigen-presenting cells, use MHC class II to present epitopes from exogenous antigens. Of course, these nucleated cells also have MHC-1 to present their endogenous antigens. Before any cell can present epitopes carried by MHC on the cell's surface, antigens have to be processed. Let's begin by looking at the processing of endogenous protein antigens. Cells make proteins in their cytoplasm. Proteins from pathogens may also be in the cell's cytoplasm. Also in the cytoplasm is endoplasmic reticulum, or ER, and in the ER's membrane are numerous MHC-1 proteins pointing into the lumen, or inside of the ER. In antigen processing, an enzyme in the cytoplasm cuts the protein into polypeptide fragments. These are epitopes. The fragments are transported into the endoplasmic reticulum through pores. Inside the ER, the epitopes bind to the MHC-1 proteins. Vesicles form from the ER's membrane. The MHC proteins, with their attached epitopes, are in the vesicle's membrane. The vesicle fuses with the cytoplasmic membrane, distributing the MHC-1 epitope complexes onto the cell surface. In this manner, nucleated cells process antigens to display on the cell's surface epitopes from every protein in the cell. Regular cellular proteins will be autoantigens. If the epitopes are from a pathogen's proteins, the immune system can recognize and attack this infected cell. Now, let's look at antigen processing for exogenous antigens. They are processed similarly, except that the antigens come from outside the cell. The endoplasmic reticulum is not directly involved. Epitopes are bound to MHC-2, and exogenous antigens are processed only by professional antigen-presenting cells, which include dendritic cells and macrophages. An antigen-presenting cell endocytizes the antigenic particle, and epitopes are released when the particle is digested in a phagolysosome. Previously, the cell has made MHC-2 proteins and put them in the membrane of a vesicle. The vesicle fuses with the phagolysosome. The epitopes bind to the MHC-2 proteins, and the vesicle, containing MHC-2 with epitopes, fuses with the cytoplasmic membrane, which results in the MHC-2 presenting epitopes on the cell's surface. Recall that only antigen-presenting cells have MHC-2 and present exogenous epitopes. These nucleated cells also have MHC-1, which displays their endogenous epitopes. In summary, Antigens are molecules that can bind to antibodies. Endogenous antigens form inside a body's cells. Exogenous antigens come from outside the body. Epitopes are specific portions of or shapes on antigens. Cells process antigens to display epitopes on MHC proteins. Processing depends on the type of antigen. Nucleated cells process endogenous antigens. Epitopes are attached to MHC1 proteins inside endoplasmic reticulum, and vesicles carry the MHC1 proteins and epitopes to the cell surface. Only antigen-presenting cells process exogenous antigens. Their epitopes bind to MHC2 when a phagolysosome fuses with an MHC2 vesicle, and these vesicles carry the MHC2 proteins and epitopes to the cell surface.